Hey folks, welcome to the first of three short lectures about animals. The kingdom Animalia, which is the largest kingdom in the Uniconta clade and our own kingdom. There's a tree of animal diversity. We're going to be talking about these major groups, phyla of animals. But first, we'll look at general animal characteristics and some evolutionary trends we see in animals. So what does it mean to be an animal? What are the major characteristics of animals? First of all, they are multicellular. So as I mentioned in the plant lecture, there are only two kingdoms that we recognize as being totally multicellular, plants and animals. And plants were, by and large, autotrophic. We are the multicellular heterotrophic kingdom. We have no cell walls, we animals. That's kind of weird. Bacteria have cell walls. Most protists do. Plants do. Fungi do. Animals, nope. We have a plasma membrane, and there's an extracellular matrix of proteins and glycoproteins, but we do not have cell walls. We make a living by ingesting and digesting on the inside of our bodies. In contrast to fungi, which are also heterotrophic and can be multicellular, they do their digesting on the outside of their body. They release enzymes and secretions outside the cell, break things down, absorb the nutrients. We ingest and internally digest. We are consumers. When you take a phylogenetic survey of animals and compare the most ancestral to more and more derived animals, there are some clear evolutionary trends. The first being the levels of organization that are present in all of life is cellular. All animals are multicellular, but the most basal animals have no level of organization beyond a bunch of interdependent cells. They can have different kinds of cells, but do not have any level of organization above that. More derived animals have collections of cells working together to perform specific functions. They have tissues. We, for example, have muscle tissues and epithelial tissue and nervous tissue. More derived animals have organs, collections of different tissues working together toward specific functions. More derived yet would be animals with organ systems, groups of organs working together, a cardiovascular system, a digestive or respiratory system. A second trend is in body symmetry. And we see here three animal types from left to right. The more ancestral animal, it doesn't have a single axis of symmetry. It is asymmetrical. The one in the middle is radially symmetrical. There are multiple cuts that would split the critter into two equal halves. The one on the right is bilaterally symmetrical. There is only one cut right down the middle that would split it into two halves, and we would call that a right and left half of the body. Here is a radially symmetrical left versus bilateral symmetrical on the right. The guy on the left is a sea anemone. The one on the right is a crayfish or a lobster. They, like us, are bilaterally symmetrical. Creatures with bilateral symmetry have an anterior and a posterior end. They have a distinct dorsal and ventral surface. Our next trend is in the number of layers of cells that form early in embryonic development. We call them the embryonic germ layers. And not germ like cooties or bacteria, but germinal layers of cells that give rise to other things. And we'll recognize two character states: having a diploblastic embryo versus a triploblastic, having two germ layers versus three germ layers in the embryo, with the latter being more derived. This illustration features major events of early embryological development in animals. Sexually reproduced animals start off life as a zygote, a fertilized egg, the one-celled diploid embryo. Following fertilization and zygote formation, the embryo experiences rounds of mitosis, where the cells divide so fast there's no time to grow in between rounds of mitosis. We call that cleavage division. So it will split from one cell to 2, 4, 8, 16, 32, 64, and the embryo will go from a solid ball of cells to the next really important thing that happens is a process called blastulation. The embryo will become hollow. All these cells, which are now small, migrate out and form a single layer on the outside with a cavity inside called the blastocele. Imagine cutting a tennis ball in half, right? And the outer wall of the tennis ball is one layer of cells, and the hollow space inside is the blastocele. In diploblastic animals, they go through a process called gastrulation, where at a certain point on the surface, cells start to push inward, invaginate, press in and form a tube that penetrates to the interior of the blastocele. That orifice, that sort of sinkhole where the cells start to pour in, is called the blastopore. And now the embryo has two layers of cells. The ones that remained on the outside forms the germ layer called ectoderm. They're blue in the illustration. And the cells that invaginated are a layer called endoderm, the ones colored yellow. And that space created by the invaginated cells forms the archenteron, which basically means the primitive gut. And that is the rudiment of the digestive system. Triploblastic embryos would form a third layer that pinch away from the endoderm and fill in some of the space between ecto and endo, and we call that third layer mesoderm. And that would make you a triploblastic organism. Our next trend is an increase in segmentation of the body. In more derived animals, the body is differentiate into segments and specialize according to segments. This lobster has a head segment and a thoracic segment and an abdominal segment, like us, right? We have a head, a thorax, and an abdomen. We are segmented. We have a series of ribs. We have a series of vertebrae. So the dividing the body up into segments allows each segment to specialize and perform certain functions and have unique anatomy. Less derived animals have less segmentation. The digestive system. One of two priorities in all living things is to find nutrition and find mates. So the digestive system is obviously a pretty important center point of anatomy, and there are clear trends in the digestive system of animals. The most ancestral animals do not have a digestive system. They draw in food particles, but individual cells trap the food particle and digest it within their cells, intracellular digestion. More derived animals have a cavity in which digestion happens, but that cavity only has one way in and one way out. It's the same way. Food goes in to the space, digestion happens, waste goes right back out through the same opening. The most derived animals have a digestive system like a tube, where food goes in one end, waste goes out the other end, and along the way, that tube is specialized. We have one, right? Our so-called gut in writ large, our digestive tube. We have a mouth and a pharynx and a stomach and a small intestine and a large intestine. So along the way, that tube is specialized to perform different digestive functions. This is one of the phylogenetic trees of animals in chapter 27 in your textbook. Almost all animals belong to one big clade, right? So this branch right here called the bilateria, that is every critter that has bilateral symmetry. Those organisms also are all triploblastic. They have an ectoderm, an endoderm, and a mesoderm. And I want you to notice how that bilateria branch has two major sub-branches here labeled protostomia and deuterostomia. So what's the difference between a protostome and a deuterostome? Well, it comes down to a couple things. The pattern of cleavage in early embryonic development and the developmental fate of that sinkhole that forms on the surface of the embryo called the blastopore, the point where cells invaginate to create the archenteron. So we're going to look at these two triploblastic clades. One is the protostomes, one are the deuterostomes. This is just a review here of these terms. We have the blastopore right here, where cells have invaginated to create this tubular structure we call the archenteron. That is the primitive gut. That's the tube that will form the digestive system. So in the protostomes, cleavage is determinate and spiral. Determinate cleavage means early in the embryo, each cell, called a blastomere, has a developmental fate. And the plane of division alternates at 90-degree angles each time mitosis happens. So the embryo appears to have cells that spiral. In protostomes, the blastopore will form the mouth, and hence the name protostome means mouth first. Deuterostomes have radial cleavage. So as the cell divides from 2 to 4 to 8 to 16, the sort of axis or plane of mitosis doesn't rotate. So the early ball of cells has radial symmetry. In deuterostomes, each cell in that ball of cells hasn't yet locked into a developmental fate. So the cells are indeterminate. And the blastopore will become the critter's anus, not the mouth. Deuterostomes. So here's a nice side-by-side with protostomes on the left, deuterostomes on the right. Look at the 8-cell stage. How in the protostome, that ball of cells has radial symmetry, but in the deuterostome, the plane of cleavage in the second round shifted so that the cells have this spiraling arrangement. We're looking at the blastopore here on the second frame, and when you follow down to the third, the blastopore forms the mouth, but in the deuterostomes, the blastopore forms the anus. This illustration also shows a difference in how the mesoderm forms, pinching away near the blastopore versus the cells that become mesoderm pinch away at the distal end of that archenteron. Our last evolutionary trend is in the body cavity. Do they have what we would call a true body cavity, an organ containing internal space? It's called a coelom. So a couple questions. Is a body cavity present? And is that body cavity lined with tissue derived from the mesoderm layer? If the answer to the first question is no, there is no organ containing body cavity present, that creature is said to be acoelomate. The prefix a is without a coelom. This flatworm is an acoelomate. When you cut it in half, the only space you see is the digestive system. Otherwise, it is completely filled with cells. There is no space in which organs are housed. That is an acoelomate animal. When you cut this roundworm in half, you see there is an internal space where it'll have things like ovaries and testes, but that space is lined only on the outer wall by mesoderm-derived tissue, in this case muscle. On the inner surface of that space, there is no mesoderm-derived tissue. So this is said to be a false body cavity or a pseudocoelom. So these animals are called pseudocoelomate. The most derived clades are coelomates. They have an organ-containing body cavity lined with mesoderm on both the outer and the inner surface. This earthworm is a coelomate. You are a coelomate. You have a thoracic cavity where your heart and lungs are. You have an abdominal cavity where your liver and intestines are. And it is lined on both outer and inner surface by a mesoderm-derived sheet of cells. So here's that tree again. In the next lecture, we're going to look at animal diversity, and most of these phyla we're going to talk about, at least the Porifera, the Cnidaria, the Echinodermata, the Chordata, the Platyhelminthes, the Mollusca, Annelida, Arthropoda, and Nematoda. Those are the major phyla, major in terms of numbers of species present, phylogenetic importance from a human perspective. There are something like 35 phyla of animals recognized, so we're not going to go through all of them, but we'll be looking at an important subset in the next lectures.